Did you know that every second there are thousands of proteins in your body that are folding into precise shapes? And even if one folds incorrectly, it can lead to a disaster. Hello, my name is Dean.
And my name is Evan, and welcome to our presentation on how this protein stability impacts the future of our medicine.
Throughout this video, our goal is to leave you with a better understanding in three main areas. First, we want you to understand how protein folding and stability determine biological function. We also want to show you how scientists study and modify proteins using experimental methods. Finally, we want to give you a better understanding on how protein structure and stability impact medicine and biotechnology. We're going to try to do this by reviewing several peer-reviewed empirical journal articles that highlight our main points.
First of all, we need to cover the basics. What is a protein and how do they function? Proteins are a macronutrient made of amino acids that act as the building block for the body. They play a pivotal role in repairing tissue and building muscle. The protein fold into specific three-dimensional shapes. This shape actually determines how the protein function in the body. So if a protein folds incorrectly or loses its stability, it may not function properly, and that can lead to disease or potentially death.
Now that we know a protein's shape determines its function, we need to understand how that shape is maintained. Protein stability comes from interactions within the protein and its environment. One important type is hydrophobic interactions. This is when parts of the protein that don't like water move toward the inside and cluster together. By staying in the center, they help hold the protein structure together and they keep it stable. Another factor is hydrogen bonding. This happens when parts of the protein form small attractions with surrounding water. These interactions help stabilize the outer surface of the protein and keep its shape intact. It's also important to understand that entire proteins don't hate or need water. Instead, different parts of the same protein behave differently. Some regions avoid water and move inward, while others interact with water on the outside. Together, this balance helps the protein fold into its correct shape. Understanding protein stability is important for several reasons. First, protein stability is directly related to proper function in the body, because proteins need to maintain their specific shape in order to do their job. When a protein loses its stability, it can begin to unfold or fold incorrectly. When this happens, the protein may no longer function properly. In some cases, this can prevent important processes from happening. This can lead to diseases such as cancer. While this may sound serious, understanding protein stability also allows scientists to design and improve treatments. By targeting specific proteins, especially those involved in disease, researchers can develop more effective therapies. In biotechnology, this knowledge is used to engineer proteins for specific purposes, such as creating more efficient enzymes, improving vaccines, and designing proteins that can function under different conditions. Now let's take a look at the research study that this project is based on. This study is an experimental biophysical investigation of protein stability, conducted by Hung and colleagues in 2021.
In this study, the researchers wanted to understand what plays a bigger role in this protein stability. They focused on two main factors: hydrophobic interactions in the protein core and hydrogen bonding on the surface. They then tested how disrupting each of these interactions affects the stability of the protein using the BIB protein as their model system. A BIB protein is part of a group of proteins that regulate when a cell should die. This is important for maintaining healthy tissues.
To study protein stability, the researchers used several key techniques. First was circular dichroism. This was used to monitor how protein folds and unfolds under different conditions. They also used electron spin resonance. This allowed scientists to see if there was any change in the protein structure and stability. Finally, they used something called the QTY code, which modifies parts of the protein to test how hydrophobic interactions contribute to stability. To test protein stability, the researchers changed certain conditions to see how the protein responded. First, they added a chemical called guanidinium hydrochloride. which disrupts interactions in the protein, especially on the surface. They also change the temperature to observe how the protein folds and unfolds under different conditions. Finally, they use glycerol, which helps strengthen hydrophobic interactions and stabilize the protein's core. The results of the study showed that the protein can unfold under both high and low temperature. They also found that while hydrophobic interactions do contribute to stability, they are not enough on their own to fully stabilize the protein. Most importantly, disrupting hydrogen bonding on the surface had a much greater effect on stability. This suggests that surface hydrogen bonding plays a more significant role in maintaining protein stability than we previously thought.
These findings are important because understanding protein stability helps improve drug design and development. By knowing how proteins maintain their structure, scientists can design drugs that specifically target proteins involved in disease. For example, targeting bit-protein interactions can be used in cancer treatment by restoring the normal process of cell death. This is important in biotechnology because scientists can engineer proteins for specific purposes, which can lead to better vaccines. This directly connects to one of our goals to help you understand that protein stability and structure impact medicine. Targeting proteins like those ones in the study are already being used in cancer treatment.
While this study helped us explore how scientists study protein stability, other research helps expand on these ideas and connect them to both biological function and real-world application. This study is titled Targeting Anti-Apoptic BCL2 Family Proteins, written by Volger and colleague. Its main focus is on BCL2 family proteins, which regulate cell death in healthy cells. This process helps remove damaged or unnecessary cells. However, in some cancers, these proteins are overexpressed, which allow for cancer cells to avoid death and continue growing. The researchers in this study have developed drugs that specifically target these proteins, which restore the normal process of cell death. This connects to our project because it shows how understanding protein structure and interactions can be directly applied in medicine, particularly in cancer treatment.
The next study is titled Principles of Protein Folding, a perspective from simple exact models by Dill and colleagues. It shows how protein folding is driven by interactions across the entire molecule, not just in one specific region. It also provides evidence that one of the most important factors in protein folding is hydrophobic interactions, which cause parts of the protein to cluster together and form a stable core. These interactions allow the protein to fold in specific three-dimensional structures, which ultimately determine its function. This connects to our project because it helps explain how the protein folding and stability directly affect biological function.
This next study is titled Two-Dimensional Electron Spin Resonance and Slow Motions. It is written by Saxena and Fried. The study explains how ESR, electron spin resonance, can be used to study protein. ESR allows scientists to observe how proteins move and change structure over time. It is especially useful for detecting molecular motion and understanding how a stable protein is under different conditions. This connects to our project because it shows how scientists use advanced experimental techniques to study protein and structure.
The final study we looked at was titled Characterization of Hydrophilic and Hydrophobic Co-shells Microcapsules, written by Hugh and colleagues. The study focuses on how hydrophilic and hydrophobic interactions can be used to control a protein structure. The researchers created core shell microcapsules, where a hydrophilic core is surrounded by a hydrophobic shell. The structure allows substances inside the capsule to be protected and released in a controlled way. The study gave evidence that adding a protein shell significantly slowed the release of compounds. This shows the structure has a direct effect on the function. This connects to our project because it shows how the same interactions that stabilize proteins can also be applied in biotechnology, especially in the drug delivery system.
To conclude, protein structure and stability play a critical role in determining how protein functions in the body. We saw that protein stability depends on multiple different interactions. These include hydrophobic interactions and hydrogen bonding. These work together to maintain structure. We also explored how scientists use experimental techniques like electron spin resonance to study protein behavior and stability. Finally, we showed how this understanding can be applied in real-world situations, which include drug design, cancer treatment, and biotechnology applications, like the controlled drug delivery. Overall, understanding protein stability allows scientists to better control how proteins behave, leading to important advancements in medicine and technology.
Thank you for listening to our presentation. We hope this gave you a better understanding of how something as small as protein structure can have a massive impact on human health and technology.